Welcome to our fourth and final lecture on clinical genetic testing. This lecture will cover specialized genetic testing. The specialized genetic testing approaches we will cover will include methylation analysis, uniparental disomy testing, expanded repeat analysis, chromosome breakage studies, telomere analysis, biochemical analysis, tumor or somatic testing, and RNA testing. First, let's discuss methylation analysis, an essential aspect of epigenetics. Epigenetics studies changes in gene activity that do not involve alterations to the underlying DNA sequence. These changes can significantly affect how genes are expressed, ultimately influencing traits and susceptibility to diseases. Methylation involves adding a methyl group, or a small chemical tag, to specific DNA bases, usually cytosine. This modification can silence genes, effectively turning them off. Methylation analysis evaluates the methylation status of specific genes or genomic regions, which is crucial for understanding how gene expression is regulated. The process begins with collecting samples, typically from blood or saliva, followed by DNA extraction. Next, the extracted DNA is treated with a chemical called bisulfite, which converts unmethylated cytosines into uracils, while leaving methylated cytosines unchanged. This step is critical and helps researchers differentiate between methylated and unmethylated DNA. After bisulfite treatment, the DNA is amplified using a PCR reaction to increase the quantity of DNA available for analysis. The amplified DNA can then be analyzed using various techniques such as methylation-specific PCR, next-generation sequencing, or pyrosequencing. These methods enable scientists to observe the methylation patterns in the genes they're studying. Abnormal methylation patterns can serve as indicators of diseases, including cancer and genetic disorders. A notable example is Prader-Willi syndrome, which occurs due to the abnormal methylation on chromosome 15, leading to the lack of expression of critical genes inherited from the father. This case highlights the significant role that methylation plays in genetic regulation and health. Uniparental disomy, or UPD, occurs when an individual inherits both copies of a chromosome from one parent instead of one copy from each parent. This phenomenon can lead to various genetic disorders depending on whether the disomy is maternal or paternal. UPD can arise through several mechanisms, including gamete complementation, trisomy rescue, monosomy rescue, and post-fertilization errors. In the case of paternal uniparental disomy, both copies of a chromosome are inherited from the father. This can lead to conditions such as Prader-Willi syndrome, which we discussed earlier. This syndrome arises when there's a lack of expression of genes on the paternal chromosome 15, resulting in symptoms like obesity, intellectual disability, and hormonal imbalances. On the other hand, maternal uniparental disomy, where both copies come from the mother, can lead to Angelman syndrome. This disorder is characterized by developmental delays, speech impairments, and ataxia. Angelman syndrome occurs due to the loss of function of genes on the maternal chromosome 15. Uniparental disomy testing begins with collecting a biological sample, such as blood or saliva, from the individual. After extracting the DNA from the sample, the DNA undergoes genotyping, which analyzes specific markers, often using SNPs. This analysis assesses allele inheritance, checking for the presence of both alleles from one parent and none from the other. Large regions of homozygosity, where both alleles are identical, indicate UPD. If UPD is suspected, additional tests may confirm the finding and explore its clinical implications. Now let's discuss expanded repeat analysis, a key technique in genetic testing for identifying specific mutations. This method focuses on a region of DNA where short sequences of nucleotides are repeated. When these repeats expand beyond the normal range, they can lead to genetic disorders. The process begins with collecting a sample, usually blood or saliva, followed by DNA extraction. The regions containing the expanded repeats are then amplified using PCR to increase the DNA quantity. Next, techniques like capillary electrophoresis or next-generation sequencing analyze the amplified DNA. Capillary electrophoresis separates the DNA fragments by size, detecting variations in repeat length, while next-gen sequencing provides a comprehensive view of the repeat regions. Expanded repeat analysis is essential for diagnosing disorders associated with repeat expansion, such as Huntington's disease, myotonic dystrophy, and Fragile X syndrome. The number of repeats can correlate with disease severity and age of onset, guiding treatment and informing family planning. Next, let's explore chromosomal breakage analysis. This analysis focuses on detecting chromosomal breakage in fragile sites, which are specific regions on chromosomes that are prone to breakage under stress or in the presence of certain agents. The process begins with collecting a sample, typically blood, and isolating lymphocytes. These white blood cells are then exposed to specific agents to stimulate cell division. Once the cells are dividing, they are treated with a chemical to halt the division process during metaphase when chromosomes are most visible. The cells are then harvested and the chromosomes are stained to enhance visibility. A cytogeneticist examines the stained chromosomes under a microscope to identify any breakage or rearrangements. This analysis can reveal critical information about the chromosome's stability and integrity. Chromosomal breakage analysis is particularly useful for diagnosing conditions such as Fanconi anemia, ataxia telangiectasia, and certain types of leukemia. These disorders may be associated with increased chromosomal fragility or breakage. Now let's discuss telomere analysis, an important technique in genetic testing that focuses on evaluating the length and integrity of telomeres. Telomeres are repetitive DNA sequences located at the ends of chromosomes, serving to protect the genetic material from degradation and prevent chromosomal fusion. They play a critical role in cellular aging and stability. The process of telomere analysis begins with collecting a biological sample, such as blood or skin cells. After extracting the DNA from these samples, scientists measure telomere length using techniques like quantitative PCR or Southern blot analysis. Quantitative PCR allows for precise measurement of telomere length compared to a reference gene, while Southern blotting provides a more detailed view of the telomere structure. Abnormal telomere length can indicate issues with telomere maintenance, which is crucial for cellular health. Shortened telomeres can lead to chromosomal instability, increased cell death, and the onset of age-related diseases. One notable example of a syndrome caused by a telomere maintenance defect is dyskeratosis congenita. This rare genetic disorder is characterized by symptoms such as abnormal skin pigmentation, nail dystrophy, and oral leukoplakia. The patients with dyskeratosis congenita often exhibit significantly shortened telomeres due to mutations in genes responsible for telomere maintenance. This defect can lead to increased risk for bone marrow failure and various cancers, highlighting the importance of telomere analysis in understanding and managing such conditions. Now let's talk about biochemical analysis, a crucial technique in genetic testing that focuses on evaluating the biochemical markers in an individual's body fluids, such as blood, urine, or cerebrospinal fluid. This analysis helps identify metabolic disorders caused by enzyme deficiencies, abnormal metabolite levels, and other biochemical irregularities. The process begins with a collection of biological samples. Depending on the suspected condition, blood or urine samples are analyzed to measure the levels of specific metabolites, enzymes, or proteins. Common techniques used in biochemical analysis include chromatography, mass spectrometry, and enzyme assays. Chromatography separates different compounds in a sample, while mass spectrometry provides precise measurements of molecular weights, allowing for the identification of specific metabolites. Enzyme assays measure the activity levels of specific enzymes to determine if they are functioning properly. Biochemical analysis is particularly valuable in diagnosing various inherited metabolic disorders. For example, it can help identify conditions like PKU, where the body lacks the enzyme needed to metabolize phenylalanine, leading to its harmful buildup in the blood. Other examples include maple syrup urine disease, which results from a deficiency in branched chain alpha keto acid dehydrogenase, and galactosemia, where the body cannot properly metabolize galactose due to enzyme deficiencies. By detecting these biochemical abnormalities, clinicians can provide early diagnosis, initiate appropriate treatments, and implement dietary restrictions to prevent serious health complications. Now let's discuss tumor testing, also known as somatic testing, a crucial aspect of cancer diagnosis and treatment. This analysis focuses on examining the genetic mutations present in tumor cells, distinguishing them from the individual's germline DNA, which is inherited from their parents. The process begins with obtaining a tumor sample, which can be acquired through various methods, such as biopsy or surgical resection. Once the tumor tissue is collected, DNA is extracted from the cancer cells. This DNA is then analyzed to identify somatic mutations, or changes that occur specifically within the tumor that are not present in normal cells. Common techniques used in tumor testing include next-gen sequencing, which allows for the simultaneous analysis of multiple genes associated with cancer, and PCR, which can amplify specific regions of interest to detect mutations. These methods help identify genetic alterations that may drive tumor growth and influence the cancer's behavior. Tumor testing plays a pivotal role in personalized medicine as it helps determine the most effective treatment options for patients based on the specific mutations present in their tumors. For example, mutations in the EGFR gene can indicate a patient's potential response to targeted therapies for non-small cell lung cancer. Similarly, identifying mutations in the BRCA1 or BRCA2 genes can inform treatment decisions for breast and ovarian cancers. By understanding the genetic makeup of a tumor, clinicians can tailor treatment plans to the individual's specific cancer profile, enhancing the chances of successful outcomes and minimizing unnecessary side effects. With that being said, let's take a closer look at cancer development. Cancer occurs when cells in the body begin to grow uncontrollably due to genetic mutations that disrupt normal cell regulation. These mutations can accumulate over time, leading to the transformation of healthy cells into cancerous ones. Several factors contribute to this process, including inherited genetic mutations, environmental influences, and random errors during cell division. As mutations build up, they can interfere with critical processes like cell growth, division, and death, ultimately leading to the formation of a tumor. One of the key models that helps explain the development of cancer is the two-hit hypothesis proposed by Alfred Knudsen. This theory suggests that for certain cancers, particularly those involving tumor suppressor genes, two hits or mutations are required for a cell to become cancerous. The first hit is often inherited, meaning the person is born with a mutation in one copy of a tumor suppressor gene, such as the RB1 gene in retinoblastoma. However, the presence of one normal copy of the gene usually keeps the cells functioning properly. The second hit is acquired during a person's lifetime due to environmental factors, random mutations, and other mechanisms, which inactivates the remaining functional copy of the gene. When both copies of the tumor suppressor gene are mutated, the cell loses its ability to control growth and division, leading to tumor formation. This model is particularly important for understanding hereditary cancers, where individuals are already predisposed to developing cancer because of the first inherited mutation. This hypothesis can also explain sporadic cancer, where there were two acquired mutations instead of one inherited and one acquired. However, this is a little bit more rare and can be described as lightning striking twice. Finally, let's move on to discuss RNA testing, which focuses on analyzing RNA, obviously, the molecule that plays a crucial role in gene expression. While DNA holds the genetic instructions, RNA acts as the messenger that carries those instructions from the DNA to make proteins, which are the building blocks of the body. DNA testing is particularly useful for detecting issues with gene expression. It can identify if certain genes are being overexpressed, underexpressed, or not expressed at all. This kind of testing is valuable for understanding diseases like cancer, where abnormal gene expression can drive tumor growth, or in conditions like muscular dystrophy, where incorrect RNA splicing can lead to improper protein production. The process typically starts with a collection of RNA from tissue samples, such as blood or biopsy material. Once RNA is extracted, it can be analyzed using techniques like RNA sequencing or RT-PCR to determine the levels of specific RNA molecules in the sample. By studying RNA, we gain insights into which genes are active and how they contribute to disease. Now, let's end this four-lecture series with a review of some important nomenclature used to describe variants. In genetic testing, it's important to distinguish between changes in the genomic DNA and the resulting protein. In terms of nomenclature, G dot represents a genomic DNA change, which refers to alterations in the full sequence of DNA, including both coding and non-coding regions. On the other hand, P dot denotes a protein change, describing how a DNA mutation affects the protein sequence. These notations are key for understanding how specific DNA mutations translate into changes at the protein level, helping to predict the impact on cellular function. Moving on, C dot represents a change in the coding DNA sequence, or cDNA. This focuses specifically on the part of DNA that is transcribed into mRNA and codes for proteins. So, C dot 76A to T would indicate a mutation in the coding region where adenine is swapped with thymine at position 76. Now let's take a closer look at how splicing mutation nomenclature works, specifically how we denote mutations at the beginning or end of an intron. When identifying splicing mutations, we often need to describe exactly where within the intron the mutation has occurred. To do this, we use specific symbols. For mutations occurring near the beginning of the intron, we use a plus sign to indicate the position of the mutation, counting from the last nucleotide of the preceding exon. For mutations near the end of the intron, we use a minus sign to count backward from the first nucleotide of the following exon.
For instance, G.75T to A refers to a mutation in the genomic DNA. This indicates that at position 75 of the genomic DNA, thymine is replaced by adenine. This could be in any part of the gene, including non-coding regions that are critical for gene regulation. On the other hand, C.73 plus 2T to A represents a mutation in the cDNA, specifically in the splicing region. The plus 2 indicates that the mutation is located 2 nucleotides into the intron, just after exon 73. In this case, thymine is replaced by adenine at that specific point, which can affect the proper splicing of the mRNA. Again, some important molecular nomenclature we see is using C dot for coding DNA sequences, G dot for genomic sequences, P dot for protein sequence, and M dot for mitochondrial DNA sequences. It's important to note that mitochondria have their own genetic material that is separate from the nuclear genome. Furthermore, we have R dot, which represents an RNA sequence. Finally, N dot is used for a non-coding RNA reference sequence, referring to genes that produce RNA transcripts but don't directly code for proteins. Each type of notation helps pinpoint the exact location and type of mutation in different molecules, allowing for precise identification and analysis during genetic testing. Continuing with molecular nomenclature, we have a greater than sign, which is used for substitution mutations, where one nucleotide is replaced by another. An equal sign represents a silent or synonymous mutation, where the change in the DNA sequence does not alter the resulting protein. Even though the DNA sequence is different, the same amino acid is produced. A star, or TER, indicates a termination or stop codon. This signifies a mutation that prematurely ends the protein sequence, often leading to a truncated, non-functional protein. Del stands for deletion, where one or more nucleotides are removed from the DNA sequence. Duped refers to a duplication, where a segment of DNA is copied and inserted next to the original sequence. Lastly, INS means insertion, where additional nucleotides are added into the DNA sequence. I'm not going to go over this entire slide, but just know that this is standard and colloquial nomenclature for CFTR mutations and variants.
The table just gives a great example of all the different kinds of nomenclature and how they're used. Let's conclude our 4-part series on clinical genetic testing by interpreting the following variants. Feel free to pause the video here and see what you can work out for yourselves. Let's consider the first variant. In this notation, C.1824 C to T indicates that at position 1824 of the coding DNA, cytosine is substituted by thymine. P.Gly608 with the equal sign means that this substitution does not change the protein sequence. Glycine at position 608 remains unchanged. This is an example of a silent mutation. Now let's take a look at this second variant. Here, C.764_766 del TCA indicates a deletion of the nucleotides TCA spanning positions 764 to 766 in the coding DNA. The PILE255 del shows that this deletion leads to the loss of isoleucine at position 255 in the same protein sequence. This is a frame shift mutation that may significantly impact protein function. Give yourselves a round of applause. This concludes our 4-part series on clinical genetic testing. Thank you guys so much for watching.